Good day, everyone. Today, we will be presenting an overview of our privately owned medical laboratory, namely Key Institute for Next Level Diagnostics, or also known as King Laboratory. Our laboratory is not affiliated with any institution and is fully capable of conducting tertiary level examinations. It occupies a total floor area of 352 square meters with dimensions of 22 by 16 meters. Within this space, we have organized several key sections, including clinical chemistry, hematology, clinical microscopy, microbiology, blood banking, immunology and serology, histopathology, and molecular biology. Each section is designed to support specialized testing to ensure efficient diagnostic workflows. Our laboratory's vision is to become the top privately held freestanding medical laboratory in the Philippines. We aim to lead a large-scale practice and research across all major areas of medical laboratory science, ranging from clinical chemistry and hematology to microbiology, immunology, parasitology, toxicology, and molecular diagnostics. Through diagnostic innovation, we help to contribute to building a healthier and more sustainable Filipino community. Our laboratory's mission is to consistently provide accurate and precise results across all essential laboratory disciplines, upholding the skills, values, and technologies required in modern healthcare practice. As a private institution, we are committed to community partnership, education, and research while maintaining high quality diagnostic testing and promoting social and health equity. Ultimately, our goal is to support lifelong professional development and improve patient outcomes as we continue to advance national healthcare.
For this part, I'll be discussing the organizational chart of a standard tertiary clinical laboratory. At the very top of the structure is the medical director, who holds the highest administrative authority and oversees the entire laboratory. Supporting the medical director are two main specialists, the anatomic pathologist and the clinical pathologist, each assisted by a pathologist assistant. Below them is the chief medical technologist, who manages the technical operations of the laboratory. Working closely with them is the laboratory manager, who helps supervise workflow, staff coordination, and overall laboratory efficiency. Under the administrative level, the lab is divided into 8 major sections: clinical chemistry, hematology, clinical microscopy, microbiology, blood bank or immunohematology, immunology and serology, histopathology, cytotechnology, and molecular biology. Each of these sections is headed by a section supervisor, who ensures that procedures are followed and results are accurate. Under each supervisor are the medical technologists, who perform the testing and analysis. Supporting them are the medical laboratory technicians, followed by lab aides who assist with routine tasks and sample handling. Overall, this chart shows how each role contributes to smooth laboratory operations, from leadership and supervision down to testing and support services, ensuring reliable and efficient patient care.
So let's proceed to the floor plan proper of our laboratory. As you can see po, yung first po na dadatnan ng mga patients ang business area. Sa business area po, andito po mahanap yung ating waiting area at yung reception. Tapos, sa reception po, of course, dito po sila magsasabi kung anong mga test yung kailangan nila, magbabayad. And then, after po nila magbayad, didiretso po sila sa dalawa po naming collection area. Isang collection area for phlebotomy at isang collection area for toilet. As you can see po, meron pong pass-through window sa likuran para easy access na po pagbibigay ng specimens para sa testing. And makikita rin po na connected sa business area is may dalawa po pong toilet, which is public access toilet po sila. So, di naman necessarily po na kailangan magpa-test para ma-access yun. Pwede pong pag naghihintay lang sila, pwede po silang gumamit ng toilet. And makikita po sa tabi ng reception ay ang pathologist office. So, ang pathologist po yung nag-supervise sa mga medtech sa lab na to. So, if may concern man po yung mga patients, ay they can raise their concern there. Tapos po makikita po, meron pa pong isang reception area. So, dalawa po yung reception namin. Yung reception po na to the left ay ang reception for blood banking. Since unlike other tests po, yung blood banking is marami pa po siyang kailangan na test bago makapag-donate ng dugo. At makikita po dito, meron pong interview room. Since kailangan po ma-interview muna ng mga nagdo-donate. And isa pa po ang screening room. So, after po nung ma-interview at ma-screening ng mga nais mag-donate ng dugo, andun na po yung bleeding area. Kung saan doon na po sila kukunan ng dugo. As you can see po, connected rin po ito sa mismong lab ng immunohema or blood banking. Para po hindi na po malayo yung binabiyahe ng mga medtech natin para ma-process and ma-store man yung donated blood. Ngayon naman po, let's proceed to the sections of the clinical laboratory.
So, welcome to the clinical chemistry section. This is where we focus on analyzing body fluids, mostly blood and urine, to check important things like blood glucose, cholesterol, electrolytes, and liver and kidney function. Basically, this section helps doctors understand what's happening inside a patient. So the whole purpose of clinical chemistry is to make sure we follow a standard, reliable procedure so that the results we release are always accurate and consistent. We deal with chemical components like carbohydrates, lipids, proteins, ions, and even some drugs or toxins that may be present in the body. Now, let's talk about the people working here. First, we have the chief medical technologist. They manage the daily operations, check if procedures are being followed properly, and make sure all results are correct before they reach the doctor. Next are the medical technologists. They're the ones running the tests, preparing samples, operating the machines, checking if results make sense, and recording everything carefully. We also have the medical laboratory technicians, who assist in preparing reagents, handling samples, helping with routine tests, and keeping the lab clean, organized, and safe. Now, let's move on to the procedures. Everything starts with sample reception and processing. When blood or urine samples arrive, we check the information, make sure everything is correct, and inspect the sample quality. For blood, we usually spin it in a clinical centrifuge to separate plasma or serum for testing. Next is quality control and calibration. Before running any patient test, we make sure the machines are working properly. We use control samples to confirm accuracy and calibrate instruments when needed. Then we go to the analysis stage. Here, we use both automated machines and manual methods. Some of the techniques include spectrophotometry, immunoassays, and more advanced method for certain tests. The main machines we use in this section include blood gas analyzer for checking oxygen, carbon dioxide, and pH. Chemistry analyzer, the main machine for most chemistry tests. Electrolyte analyzer for sodium, potassium, chloride, and other ions. Spectrophotometer for measuring chemical concentrations using light. Water bath for keeping reagents or samples at a stable temperature, and also refrigerator for storing reagents and some patient samples. After the analysis, we go to do result reporting and validation. The medical technologist reviews the results, checks if they're accurate, and once everything looks good, the results are forwarded to the doctor. And of course, safety measures are always followed. We treat all samples as potentially infectious. Wearing a lab coat and gloves is required, and eating or applying makeup inside the laboratory is strictly not allowed. And that's the clinical chemistry section, organized, precise, and essential in helping diagnose and monitor different health conditions.
Hematology section and give you a quick overview of how we handle and process blood samples. Hematology focuses on the study of blood, blood-forming organs, and blood diseases. This is where common tests like CBC or complete blood count, hematocrit, and hemoglobin are being performed. Our SOP is designed to make sure all procedures in this area are being done safely, accurately, and consistently from the moment the sample arrives until the results are being released. The purpose of our SOP is to guide in handling, testing, and reporting blood samples the right way. This helps us detect blood disorders and bone marrow diseases accurately. which is essential for proper diagnosis and patient care. The scope includes all the steps in the hematology workflow: receiving, preparing, analyzing, and reporting samples. It applies to all staff since hematology testing is used to identify blood problems, certain cancers, and inflammatory conditions. In terms of responsibilities, the hematopathologist oversees the section and interprets complex cases. Medical technologists run the tests using machines like automated hematology analyzer, tube mixer, slide snader, and microscope. Technicians help with the specimen preparation and quality checks, while phlebotomists collect properly labeled EDTA blood samples. The chief medtech supervises the whole workflow. For the process itself, blood is collected in EDTA tubes. It is gently mixed and brought to the lab immediately. They reject samples that are clotted, hemolyzed, or mislabeled. Of course, before running inpatient care, daily quality control is done. Automated results are validated and anything abnormal is confirmed through a blood smear under the microscope. Results are entered into the LIS and any critical values are reported right away. Through the entire workflow, they follow strict safety guidelines using PPE, disinfecting work areas, maintaining equipment, and properly disposing of waste. That is all for hematology section.
In the clinical microscopy section, our main purpose is to provide accurate and reliable results from body fluid examinations, including urine, stool, and other specimens. This section covers all personnel who collect, label, and analyze samples to help ensure proper patient diagnosis. The chief medical technologist supervises the entire section and checks the result before sending them to the pathologist. The medical technologists perform the actual analysis under the microscope and make sure proper procedures are followed. The assistant medical technologists receive and label the specimens and handle the cleaning and sterilizing of equipment to avoid contamination. We use several important instruments: 2 clinical centrifuges for preparing urine sediments, 2 microscopes for examining cells, crystals, parasites, and bacteria, a hemocytometer for manual cell counts, a urine analyzer for automated chemical testing, and a strip reader for accurate dipstick reading. The procedure starts with proper collection and labeling of specimens, making sure they are fresh and clearly identified. Next is documentation, where we check the volume, labeling, and any contamination before encoding the details into the system. We then perform a physical exam, observing the sample's color, appearance, odor, and noting any abnormalities. For the chemical exam, we use dipsticks or run the sample through the urine analyzer or strip reader to check for glucose, protein, bilirubin, pH, and ketones. For the microscopic exam, urine is centrifuged and stool is smeared on slides using the microscope. We identify cells, bacteria, crystals, and parasites, and compare the findings to normal values. The hemocytometer may be used when counting cells. After all tests, we validate the results, compile them, and ensure everything is accurate before the chief medtech and the pathologist give the final approval. We end with quality control, checking equipment and reagents, and with proper waste disposal, separating biological and chemical waste correctly. Throughout the process, proper protective equipment must always be worn and any accidents should be reported immediately.
In the microbiology section, it focuses on detecting and identifying disease-causing microorganisms such as bacteria, fungi, parasites, and sometimes viruses. The microbiologist or pathologist is the one who supervises the entire microbiology section, ensuring that procedures, safety practices, and quality standards are strictly followed. The medical technologists naman are the ones who perform the actual microbiology work. They culture specimens, examine colony growth, stain samples, conduct biochemical tests, perform antibiotic sensitivity testing, and record all steps and results properly. Meanwhile, the laboratory supervisor makes sure that everyone follows the SOP and safety rules and also ensures equipment and reagents are working and calibrated. The machines used in this section are clinical centrifuge, drying oven, electric or gas stove, incubator, refrigerator, waste bin, and water bath. The tests performed naman are gram staining, biochemical test, culture and sensitivity, and antibiotic sensitivity testing or AST. So first, we start by collecting the specimen. We start by checking if the sample is properly labeled and collected the right way. If the specimen is leaking, contaminated, or doesn't have any label, it will be rejected. Only the acceptable samples are logged in the system so we can track them properly. Next is inoculation and culture. Using the sterile technique, we streak the specimen onto the correct culture media. After that, the plates are incubated at around 35 to 37 degrees Celsius for about 18 to 24 hours so microorganisms can grow. Then, we move on to examination and identification. After incubation, we look at the colony stat crew, checking their color, size, and other characteristics. We also perform gram staining and basic biochemical tests like catalase, oxidase, and coagulase. These tests will help identify what organism is present. Next step is antibiotic sensitivity testing or AST. Here, we prepare a standardized bacterial suspension and spread it onto the Mueller-Hinton agar. Then we place antibiotic disc on the surface and incubate the plate again. After incubation, we measure the inhibition zones to know which antibiotics the organism is sensitive or resistant to. After that, we go to reporting. We record the identified organism and its antibiotic sensitivity pattern. If multi-drug resistant organisms is detected, it will be reported as soon as possible. Another important part is quality control. Every day, we use control strains to make sure our tests are working correctly. We also check if all reagents, media, and equipment are in good working condition. And lastly, waste disposal and decontamination. We always disinfect our work areas by following biosafety rules. This helps keep the lab safe and prevents contamination. Throughout the lab work, everyone wears gloves, lab coats, masks, and goggles to stay safe. Hands are washed a lot and work tables are cleaned with disinfectant regularly. If anything spills or someone gets exposed to germs, it is reported right away. Sharp things like needles are thrown in special containers, and warning signs are put up in areas with biohazard materials. Doing all this helps keep both the staff and samples safe.
So for immunohematology or blood banking, this section is responsible for collecting, storing, and testing blood to ensure that it is safe and compatible for transfusion to patients. So for the machines, these are the immunohematology centrifuge, refrigerator, and platelet agitator. For tests performed, blood typing, ABO and RH typing, cross-matching, antibody screening and investigation, direct antiglobulin tests, and donor screening tests. So for the standard operating procedure, the purpose is to make sure blood is collected, processed, and tested properly and that all patient results are accurate. So for the scope, it applies to all laboratory activities and blood banking personnel in handling each blood collection and to ensure that the safety of the patients. So for the responsibilities, the first one, the blood banking medical technologies. They examine and test each blood sample by following proper safety procedures to avoid any contamination. Next, the pathologies or blood banking head. They review and analyze the test results and supervise medical technologies all throughout the steps. For the procedures, blood banking involves several steps to keep blood safe for patients. First, the donor is checked to make sure that they are healthy and allowed to give blood. The blood is then collected using clean tools and handled carefully. After collection, it is processed by mixing it with an anticoagulant, separating its parts, and labeling it properly. The blood is also tested for its type, checked for antibodies, and screened for infection to make sure it is safe. All details are written down and the blood is stored correctly and used in the right order. So before transfusion, the staff double-check the records and match the blood for the patients. Throughout the process, workers use PPEs, check their equipment, and throw away used materials properly to keep everything safe.
The immunology and serology section exists to ensure that tests related to the immune system are conducted with precision and safety. Its purpose is to provide dependable results that support accurate diagnosis and patient management. The scope of this section covers all activities involving the handling, testing, and reporting of immunology and serology specimens, making sure that every step follows a standardized and reliable workflow. All personnel assigned here are expected to perform procedures consistently and maintain strict quality practices. Work begins with specimen collection, where blood samples are taken using sterile techniques and allowed to clot before separation. Once centrifuged, the serum is transferred to correctly labeled containers. Before testing starts, each sample is checked for proper patient identification and acceptable quality. Samples that need immediate analysis are kept refrigerated, while those for delayed testing are stored frozen to preserve stability. The section performs various tests that detect antigens and antibodies linked to specific diseases. Rapid diagnostic kits are used to screen infections such as hepatitis B, HIV, and dengue. These tests typically display colored lines that indicate whether the target antigen or antibody is present. For typhoid screening, the Weidel test is applied by mixing patient serum with known antigens and observing for visible clumping. Pregnancy screening is also handled here by detecting HCG in serum and urine samples.
Medical technologists conduct these procedures and ensures that each step follows established protocols. The pathologist supervises the entire process and validates results before they are officially released. All outcomes are documented in the laboratory logbook or information system to maintain complete and accurate records. Quality control is a routine requirement. Control samples are processed to confirm that reagents and test kits are functioning correctly before any patient sample is handled. Waste generated throughout the day, where including of used cassettes, gloves, and other disposable is discarded in designated biohazard containers following proper segregation rules. Safety is upheld through consistent use of personal protective equipment, also known as PPE, regular disinfection of work areas, and immediate reporting of accidents or spills.
And now, finally, in the histopathology department, we look at cells and tissues under a microscope to spot diseases like cancer. And we do tests like pap smear, fine needle biopsies, and we also fix, slice, and stain tissue samples. Our goal is to make sure that these steps are done correctly, safely, and in a consistent way. And the ones responsible for these procedures are the lab technicians, pathologists, and their supervisors. So the technicians are the ones responsible for labeling, preparing, and staining the samples. While the pathologists naman, they examine them under a microscope and they write the final reports. The supervisor then makes sure that the quality standards are met and the equipment is properly working and the staff they follow the correct process. And when a sample arrives, we check the patient's information and label it correctly to avoid mix-ups. We also record its condition, whether it's like clean or not, like how much there is, and we note descriptions like color, smell, volume, and for fluid samples naman, we test for things like protein, glucose, and pH using dipsticks. And we write down and yun nga, the results. After testing, the results are reviewed by the senior staff to make sure that they're accurate and precise. We keep track of the quality by regularly checking our equipment and reagents. We also throw away waste properly, especially the ones that are hazardous and yun nga, chemical waste. Safety is a big deal nga kasi everyone must wear protective gear like correct PPEs and any accidents are immediately reported. And and now for na naman yung tissue samples. So for the tissue samples, we put them in a fixative right way to prevent them from degrading, like yung usually formalin. And then we process them by removing water, clearing, and embedding them in paraffin. We slice the paraffin blocks into very thin pieces, like 3 to 5 micrometers. Put them on slides and then stain them. Usually with H&E. And if needed naman, we use special stains or antibodies. And then finally for the cytology samples like fluids, we make smears or use cytocentrifuge, and we fix the cells quickly and stain them. Like yun nga, pap stain. And then pathologists then they look at the prepared slides, notes what the cells look like, identifies any problem, and yun nga, finally they then write the results in our systems, verify them, and then they make issue signed reports. We also run controls for special stains and dispose all of the waste following safety rules. And now, Chyan, take it away.
So for the last part, we have the molecular biology department. So in the molecular biology department, we work with genetic materials like DNA and RNA. These are to detect the infections, the inherited disorders, and also monitor specific diseases like cancer. So we perform specialized tests like these examples could be PCR or LCR and next generation sequencing. The section focuses on analyzing the genetic material with high accuracy and also the sensitivity. So our SOP's goal for this is to ensure that every molecular test is performed correctly, reliably, and without the contamination. So this procedure applies to all the staff that are handling the molecular specimens, which can include the laboratory technologists, the analysts, and the molecular pathologists. So for the technologists, they are the ones responsible for receiving the samples, extracting the nucleic acids, and also running PCR processes and documenting findings. Um, for the molecular pathologists, they're the ones who can review, interpret, and also validating the results before it gets released. So the supervisor naman, they're the ones who ensure the quality standards are actually followed and all the equipment such as the PCR machine and the computer system is actually functioning properly. So an example is when a sample arrives, they're going to check the patient details, label the specimen correctly, and also make sure that it was transported at the right temperature. So this is so we can prevent nucleic acid degradation. So the samples like swabs, blood, or also clinical materials are recorded and stored properly. So during nucleic acid extraction, we use RNAs and DNAs free tools and follow validated methods to isolate the DNA or RNA, which are then stored to negative 20 degrees Celsius to negative 80 degrees, depending on the type. So for the PCR testing, we prepare a reaction mix which contains the template DNA, the primers, enzymes, and also the reagents. So the sample would be placed in the thermal cycler so it can undergo amplification and the results would be detected through fluorescence. We always compare each test with positive and negative controls to ensure that the run would be valid. If the controls fail, the entire test would be repeated. All results are encoded in the laboratory information system and reviewed by our senior staff. So for the quality control, it is maintained by routinely checking the equipment, documenting all the control outcomes, and ensuring that the reagents are within the proper conditions at all times. So waste materials like used tubes, pipette tips, and contaminated consumables are disposed of and designated biohazard containers. So safety would be a major priority since staff would need to wear complete PPE at all times, work in designated clean and amplification areas to prevent contamination. Uh, we would also be following BSL2 or BSL3 guidelines. Any spills, accidents, or contamination issues would be reported immediately. So after testing and verification, results would finally be finalized and validated by the molecular pathologist. This would then be released through our system. So the department would be ensured, it will ensure that all procedures from specimen handling to data interpretation would be carried out safely, consistently, and with the highest level of accuracy expected in molecular diagnostics.
Since tapos na po mapakita and explain ng mga sections dun sa clinical laboratory, mag-proceed naman po tayo sa mga staff area natin. So sa staff area po, meron po tayong hand washing area near the door, and emergency shower just in case na mangailangan man sila nun. Meron rin tayong staff pantry where staff can rest and enjoy their time while on break. And meron rin tayong mga storage area. Dito nakalagay ang mga spare equipment na hindi naman sensitive sa storing environment nila. Meron din tayong female locker and male locker para dun nakikip ang mga gamit nila at kung gusto man nila magpalit ng damit or anything. Connected yung mga locker na to sa mga gender-sensitive na staff toilet. Dito rin makatatagpuan yung service entry at saka yung fire exit. So, ito yung mga tutukuyin mamaya sa emergency preparedness. Tapos, pagkalabas mo doon, andun ang waste holding area. So, lahat ng waste na nakokolekt sa laboratory ay dito nilalabas. At merong sink sa tabi ng mga trash cans para after disposal ay makakapaghugas sila agad ng kamay. So, since nalaman na po natin yung mga standard operating procedures for each section of the laboratory, alamin naman po natin kung anong gagawin sa lab during an emergency. So, emergency preparedness. So, workers within the laboratory are routinely trained and participate in regular drills in which they are taught standard emergency procedures for the following, like fires, emergency evacuations, and earthquakes. So, regular po silang natuturuan neto. So, pagkaka-hire po ng isang tao, ay tuturo po sa kanya to and throughout the year, magkaka-periodic drills po sila. So, ano po nga ba ang kailangan nilang gawin? For example, in the case of the fire, tinuro po sa staff na kung saan ang location ng mga fire alarm, ang mga fire extinguisher, at kung asan ang mga emergency exits. So, kung sakaling may nasunog man, instructed ng staff na activate ang fire alarm and immediately evacuate the building na. In the case of emergency evacuations naman, essential kasi na since meron nga mga harmful biological agents dun sa loob ng lab, is kailangan nilang i-secure to bago sila umalis. So, kailangan nilang itigil lahat ng ginagawang test ng mga chemical processes, tatakpan lahat ng chemicals, lahat ng viruses or bacteria man na ginagamit nila, unplug nila yung mga hindi naman essential na electrical equipment kagaya ng ovens and hot plates, tapos off nila yung gases and water, tapos fully close nila yung fume hood and biological safety cabinet since alam nga natin na andun yung mga harmful biological agents. Tapos, kailangan na rin securely close all the refrigerators and freezers. Take note na hindi nila bubunutin yung mga to since kailangan ng temperature control para sa mga laman neto. So, isa siya sa mga essential electrical equipments. Pag earthquake naman, staff are instructed to, of course, duck, cover, and hold until tumigil na ang pag-shake ng ground. After this naman, staff are instructed to exit the building through designated emergency exits na since naturo nga sa kanila before.